Okay, and as Dr. Berry mentioned, we're from the Department of Environmental Science, and I'm going to start you off, in case you're not really familiar with science stuff, with what lichens are. And I've got a picture here. This is going to be in the background of every slide. And the main one that you see here is a species called Parmatrema perforatum, which is Parmeliaceae family. And the common name for that is perforated ruffle lichen, because these discs are oftentimes perforated. And you can see up here, these little discs are called apothecia. They contain spores. These lichens here, the yellow ones, those are Usnea strigosa. That's a different species. This is a different species of Granulina. So you've got about 3 or 4 different species in here. And these little fibrils and other tiny parts of the lichen will break off and reproduce asexually. That's the most common way that they reproduce. So they're pretty exciting to study, lots of different shapes. And most of these are about 1 centimeter across. So you have to really look closely to see the fine details that they contain. And one of the other things that they do is they accumulate a lot of gases from the atmosphere. The ones that I've got listed up here, sulfur dioxide, ozone, nitrogen dioxide, these are all dangerous to human health. And so we can collect the lichens and measure what they've collected over time. They're pretty slow growers, so you can get kind of an estimation. And previous research that's been done in the field of lichens has focused on the species diversity, which would be the floristic mapping there, and trace metal analysis, which is what we decided on because so little research has been done so far. In fact, there's never been a kind of lichen monitoring in Nacogdoches so far. And so there wasn't any field guide. We had to basically start off with just finding out what were the most common species around. So we went to the SFA, Bottleneck Hardwood Forest, just north of the commuter lot. We collected 23 samples, and we measured the different features, the lobes, etc., and we used an identification guide. And we came up with the one that I mentioned, the Parmatrema perforatum. We decided that it was the easiest to remove, and also we thought we would be able to find it in the most places because it was the most common.
All right, but to do that, to collect those and determine which species we were going to use, we first had to go out into the field, actually find the university commuter lot, into the bottomland hardwood forest there, and collect lichen species just to see what was out there. And this is Mark Nozel over here examining one of those. We went out and collected them and put them in acid-free paper packets so as not to damage the species at all. And we took them off with palette knives and common kitchen knives, instead of taking them off with our fingers, and we took them to the lab. This is a lab in the Arthur Temple College of Forestry building. It's an environmental science lab. And to identify, we used multiple instruments, but the main 3 rather, is this digital caliper here, and a VMR, VistaVision dissection microscope, very classy stuff. And also this right here, which is our holy grail of ID, which is the Lichens of North America ID key. All of that took place in the lab. It took a couple of hours to research through the key, and we came up with our most common species, which Jeremy referred to earlier as the perforated ruffle lichen.
And we had to determine how much we were going to pick up when we got out there. And so we took the samples that we collected already, we dried them out, and found out what we measured the area of them, and then dried them out, so we knew how large of a sample that we were going to have to create or to collect. And we used the same situations that we're going to be using later, 105 degrees Celsius oven for drying.
Okay, so to determine where we were going to collect our lichens, we looked up our lichen samples. We looked up the topping corners in the Nacogdoches area, and we took the top 15, and then we plotted them in a map, along with high traffic areas in Nacogdoches, which included like North Street, Walmart, and downtown Ag. And from these, we derived transit lines in order to properly get the scope that we wanted to ensure diversity. We started from the center of Nacogdoches and worked our way outwards. And then to add on to that, we used the windrow plot, which you can see right there on the left. A windrow plot is basically a compass that shows wind direction and like the frequency of wind. And then we went out and we got samples from where we plotted everything again. We took background samples as well as samples in the city limits, and we were careful to make, to collect samples 1 mile from major highways in order to ensure we got proper background samples. We took 24 samples within 2 weeks. We then dried these samples, and then we grinded them with a coffee grinder, dried them again for 12 hours at 105 degrees Celsius to get the dry mass for each sample. And then each sample was digested in nitric acid, which was then later used in an ICP spectrometer. A standard was created in order to compare the concentrations of the metals between the samples we collected. The metals we focused on are vanadium, chromium, copper, zinc, and lead. And what the spectrometer does, they shoot heat waves that treated the metals, and then the different metals refracted different wavelengths, and that's how we were able to determine which metals were which. And then we compared them to the standards we made in order to create our results.
So all of this sounds really cool, really fancy, but unless you can see it and unless you can understand what it means, it's really not worth a whole lot. It's just a lot of numbers on paper. And so to enable us to see a visual representation of our results, we use spatial analysis. Every time we went to a plot point over that 2-week period, we collected a GPS point, longitude and latitude, and into a device. And so we had an ID number and we had a bunch of longitude and latitude points. But that doesn't mean anything if you can't see it. So we paired it with a GIS in order to establish meaning. And a GIS is essentially a software program that helps you manipulate and display data. And most commonly seen as mapping, and so that's what you'll see on the next slide. But the maps generated from the results enabled us to link data with those points and where they were. So you'll be able to see the transect lines that were used, and also the vanadium pollution and the site index number based on a color scheme.
All right, and this is one of the beautiful maps that Whitney created in the SFA GIS lab. And you'll notice this is, we used vanadium for this map, and it's a really good indicator. It kind of shows how you've got these hot colors in the middle with high concentrations, and then you've got these low colors outside. Of course, this is over by the airport. But you've got these low colors. This is our lowest concentration right here, and it was just across the street from a watershed area. Whereas these higher ones here are, closer to the city. I'll talk about those in a second here. In order to compare, we created this formula which subtracts the baseline values, so you're only comparing what is over the baseline. And this is the graph of that you've got vanadium, copper, chromium, zinc. Now all these are in the same order. So you can see if something's high in vanadium, it's also going to be high in chromium, copper, and zinc typically. You can't really see these that well, but it's a good comparison to show that they kind of follow the same trends. And we subtracted out the baseline values. But these samples, numbers 11, 12, and 13 were the worst ones on just about everything. And then we averaged it all together to create the site index number, which is in this slide graphed out. And so this was the average of all of our research. And sample 19 we skipped because it was so high we considered that there was probably air involved. Sample 13 was North Street. Sample 11 was North Beach and Nivco. Sample 12 was north of the industrial area by Pilgrim's Pride, all the way down to the Loop. And the three lowest, of course, as I said, were outside of city limits. And the trace metal analysis showed that not only is sampling possible, but it's easy to find the necessary amount throughout Nacogdoches. And so we successfully achieved our purpose to measure and compare the trace metals of local lichens. And we think that the next step in the process should be a large-scale monitoring program using the research that we've gotten so far to map pollution trends, especially for the lead, just because it's a NAC's criteria pollutant. And once again, I'd like to acknowledge Dr. Cheryl Perez and also Mr. Wayne Weatherford, who is the lab coordinator at the Stephen F. Austin State University Soil and Plant Water Laboratory. Y'all send your samples over there. thank you very much.